Good evening. I'm Nicholas Don Richards, as I've just been introduced. I'm studying for my PhD at the University of Reading, and the topic of my presentation is the venom of the Poecilotheria regalis spider and the effects of its venom on skeletal muscle and its stem cells. Yes, okay. So firstly, some brief background information about the spider. It's the Poecilotheria regalis spider, which is also known as the Indian ornamental tree spider, and which is a member of the Therospodidae family or tarantula spider. It's found in India and is also kept as an exotic pet in other countries. So in India, it's reported that herbal treatments are used in the case of its bites due to the inflammation they cause. Symptoms include pain, symptomatic muscle cramps, and rhabdomyolysis, as indicated by plasma-creatine kinase at 370 units per litre, which is increased from a reference range of 0 to 200 units per litre. And we know literature reports that in mice, IV injection results in an LD50 of 9 milligrams per kilogram, and tonic chronic seizures and hyperalgesia, which is sensitivity to pain. There is no current antivenom. Medical treatment currently focuses on symptom management, using calcium, now using magnesium to counteract calcium. The venom is novel and characterization is required to develop more effective treatment. So the question I am asking is, what are the local and systematic effects of the venom on skeletal muscle? To begin to answer that question, a short introduction on myogenesis and what forms skeletal muscle. So skeletal muscle is produced via myogenesis using stem cells known as satellite cells, which are normally found quiescent under the basement membrane or basal lamina, and these are activated via signaling from fibroadipogenic progenitors. Satellite cells then divide and proliferate and migrate. They then fuse into, they then become cells known as myoblasts, which are progenitor cells, which are mononuclear. These then differentiate and fuse together to form multinucleated myotubes. And in vivo, these then go on to form multinucleated myoblasts, which become myofibers. Thus, these give an innate regenerative capacity to muscle. Myotubes and fibers can also undergo atrophy, where the rate of breakdown of proteins exceeds that of the formation, which results in a decrease in area of the myotubes. A venom acting on any of these processes can inhibit these functions, and so these were studied in vitro using mouse myoblast cell line, which is known as C2C12 cells, and in vivo in mice. So, beginning with proliferation and migration of the cultured C2C12 myoblast cells, I used an MTS absorbance assay, which assesses metabolic activity of mitochondria, indicated by the proliferation of colored formazan. So no significant change was indicated. So no change was seen in proliferation up to 100 micrograms per ml of venom, which is supported by imaging of the cells. And similarly, no significant change was seen in migration of the cells over time, over a 24-hour period, so that myoblasts were not affected by the venom. However, following this, myoblasts were then cultured to confluency, and they were encouraged to form myotubes in the presence of different concentrations of venom over the course of 5 days to study the effects on fusion of the myoblasts into myotubes. This showed a significant impact on the fusion index, which is the percentage of myonuclei present in myotubes compared to total myonuclei. Also, the atrophy was also impacted with significant change, or significant decrease in area at 1 and 10 micrograms per ml of venom. So this was performed on mature myotubes, and then followed a 24-hour period of venom exposure on mature myotubes. So while so differentiated myotubes were affected by the venom. Following these findings, in vivo experiments were performed using C57 Black 6 mice, which are genetically normal, to understand the effects of the venom in the context of regeneration and the systems present in an organism, such as the immune system and regeneration. To do this, the muscles were dissected at day 5, day 10, and day 20 for 3 different time points, and the muscles were weighed, and a significant impact and decrease on muscle weight was seen in the injected muscle, which was injected with 25 micrograms per ml of the venom. So a significant impact was seen at day 5 and at a reduced but still significant impact at day 10. However, by day 20, this impact was no longer significant, indicating regeneration over time. This was supported by histological analysis and immunohistochemistry using, so at day 5, there was a large amount of infiltration, so damage at day 5. This was accompanied by a high amount of fibrosis, so that's collagen being formed. So that's also present was IgG infiltration and a decrease in the intensity of dystrophin, which indicates a decrease in the membrane integrity. However, myosin heavy chain 3, which is a sign of regeneration, is present at day 5 at 100% of the fibers that were measured, showing that however, at day 10, regeneration had progressed, as shown by centrally located nuclei on the H&E stain, a reduced amount of picoceres, a reduced amount of fibrosis, and an absence of IgG infiltration, and an increase in dystrophin intensity. The myosin heavy chain had, however, was still present at 45% of the measured fibers, which indicating that while regeneration has progressed, it is still not completed. A similar story at day 20, reduced, still centrally located nuclei, so further progressed regeneration with reduced, however, the fibrosis was still present at day 20. So another area of analysis that was done on the histological samples was focusing on blood clotting and the activation of platelets and how this would impact indirectly onto the muscle and would cause thromboinflammation and perhaps a reduction in oxygen available to be delivered to the muscles. So the p-selectin indicates microthrombi present in the muscle tissue surrounding muscle fibers, which was present at day 5. However, it was decreased at day 10, although it was still present, which meaning that the microthrombi had not yet been fully resolved. This is also the case with fibrinogen, which was also increased at day 5, but reduced. However, this was reduced, but not fully absent at day 10 in percentage area. So in conclusion, the venom of the Pinthelia regalis does not affect myoblast proliferation and migration, which meaning that myoblasts can survive and function. However, myotube fusion is inhibited and atrophy is induced, which means that the P. regalis venom does have effect on differentiating and differentiated myotubes. In vivo, damage is occurring at day 5, which indicates cytotoxicity of the venom and attenuated regeneration, microthrombi and clotting, which indicates indirect damage. So regeneration is occurring at day 5, showing that damage is transitory and regeneration can occur. However, it is attenuated and the presence of fibrosis still indicates that it is not yet completed at day 20. This can be, so the migration is down to the ability of the myoblasts, the stem cells, to function. So in terms of my future work, I will continue doing histological and immunohistochemical studies. I will seek to narrow down the gap in significance between in the case of the fusion and atrophy, and gain a more detailed concentration curve there. I will also use future histology, so looking at inflammation, particularly the stain F480, which indicates the presence of macrophages in histological stains, and the laminin, which will indicate any damage to the extracellular matrix, which is the support and structural surroundings and environment of the myofibers. I will also do further systematic effects, further systematic studies on platelets and blood. So that's platelet activation, clotting, and clot retraction. So this, I will also characterize genetic expression and protein regulation changes within myotubes to find what the venom is doing on a genetic level using mass spec and microarrays. We've also fractionated the venom, and we will be studying this and its individual activities, perhaps also supported by mass spectrometry. One final question to ask is whether inhibitors can be found to reduce the effects of the venom that we've found. That only leaves to thank my supervisors, Professor Ketan Patel and Professor Shikta Valapuri, who you'll be hearing from later in this conference, and Steve Trim at Venom Tech, who supplied the venom as a lyophilized powder that I have been using. Thank you.
Unless there's a very quick question, we've run out of time for questions for this one. No very quick ones? You have a very quick one. I knew you'd want to ask.
Yeah, hi Nicholas, thank you so much and congratulations. You've looks like done an awful amount of lab work for this so far. Just out of interest, very quickly, because it's such a popular pet, I didn't realize that it had such a nasty bite. And out of interest, is this you see the sexual dimorphism in the colors? Do you think you might see sexual dimorphism in the venom as a result?
Yes, so other studies have certainly focused on only the female venom. So there is sexual dimorphism among the species. Yes.
Might you be investigating that?
Yes.
Talk to Steve. These spiders give heaps of venom. They're really good milkers, so he should be able to get you both, male and female.
Thank you so much.